Morning everyone. This is JIAS Academy, Rambaram Chennai. We are giving online and offline courses. Helpline is given here CSIR NET, Life Science, Unit 8, Part 1. This class is about Mendelian genetics in Unit 8. Let us see the first point. What is genetics? Genetics is the study of heredity. It's a study of heredity, and the father of genetics is Johann Greger Mendel. His lifespan is between 1822 to 1884. He set the framework of genetics long before the chromosomes or genes had been identified. In 1865, Mendel presented the results of experiment with nearly 30,000 pea plants to the local Natural History Society. In 1866, he published his work, Experiments in Plant Hybridization. Experiments in Plant Hybridization. In the Proceedings of the Natural History Society of Brunn. Mendel's seminal work was accomplished using the garden pea plant, which is called as Pisum sativum, to study the inheritance. You can see this is the pea plant, this is the violet flower, and this plant bears the white flowers. He crossed these two plants, and the result is all the plants, they bear violet flowers. This is in the F1 generation, and in the F2 generation it is 3:1 ratio, that is 3 parts are, that is, if you take 1,000 flowers, 1,000 plants in the sense, 705 are bear violet flowers and 224 they bear white flowers. So this is the result he got in the F1 generation. All the plants, they bear violet, and in the F2 generation it is in 3:1. And there are he has taken another 7 very important characteristics or the traits, different traits in pea plant. He selected only the pea plant and he took 7 different traits in them. The first one is flower color, second one is flower position, that is axial versus terminal. The flower color is violet versus white, and third one, the plant height, which is tall versus dwarf. Seed texture, round versus wrinkled. Seed color, yellow versus green. And pea pod texture is inflated versus constricted. Pea pod color, it is green versus yellow. So these are the main 7 different traits he has selected, and he crossed violet versus white, axial versus terminal, tall versus dwarf, round versus wrinkled, yellow versus green, inflated versus constricted, and green versus yellow. What is the result he got is, in the F1 offspring, the traits are 100% violet, that is, the violet is dominant over white. Then the axial flowers he got, that is 100% he got axial only. So the axial is dominant over the terminal. Then the third trait, that is tall versus dwarf, he got 100% tall plants. That means the tall is dominant over dwarf. Then 100% round. So from this we understood that round is dominant over wrinkled. And the color, seed color, he got 100% yellow. So this yellow is dominant over green. And the next one, pea pod texture. Pod texture in the sense, this is the part and in which the pea seeds are arranged. So the part texture, what is the texture of this part is, he has selected inflated versus constricted, and 100% inflated he got it, and green versus yellow, in that 100% green. Okay, the pod, pea pod color, this is seed color, so don't confuse that. This is pod color. Part color it is green, seed color it is yellow. So seed color yellow is dominant, part color green is dominant. Okay, so this is the result he got in his experiment. And the F2, the trait ratio is all 3:1. All 3:1. This is approximately 3. Here also approximately 3. Here also approximately 3. So these are the 7 different characters or 7 different traits he has selected from the pea plant. See, next if you see, the genetic factors proposed by Mendel were carried on. Chromosomes was proposed in 1902 by Walter and Sudan and Theodore Bory. And they decided that they concluded the theory as chromosomal theory of inheritance. The theory is called as chromosomal theory of inheritance. It is proposed in 1902 by Walter and Sutton and Theodore Bowery. And different versions of genes are called as alleles. What are allele in the sense? Different versions of genes are called as allele. Diploid organisms that have two identical alleles of a gene on their two homologous chromosomes are homozygous for that trait. Diploid organisms that have two different alleles of a gene on their own homologous chromosomes are heterozygous. That is, if you take the chromosome, in that if you take a gene, in that gene, I am just expansion this one and I am showing this is the gene in the sense. There are two alleles. That is, for example, capital Y, capital Y. So this is called as homozygous. If the alleles are different, that is capital Y and small y in the sense, this is called as heterozygous. Okay, so that is what the diploid organisms that are two different alleles of a gene on their own homologous chromosomes. Homologous chromosome in the sense, one is from mother and one is from father, or one is from male or one is from female. Okay, this is from male and this is from female in the sense. These two chromosomes are called as homologous chromosomes. Okay, and the gene which is present inside, and you can see the alleles. These two alleles are homozygous. These two alleles are heterozygous in genotype. You can see there are some examples of dominant and recessive traits in human. That is achondroplasia. This is dominant trait, and albinism is recessive trait. Brachydactyly is dominant trait and cystic fibrosis is recessive. Huntington's disease, that is dominant trait, and Duchenne muscular dystrophy, that is recessive. Marfan syndrome, that is dominant trait. Galactosemia, it is recessive. Neurofibromatosis. Phenylketonuria, this is recessive trait. Widow's peak, dominant trait. Sickle cell anemia, which is recessive. Woolly hair, which is dominant. Taysha's disease, that is recessive. So these are some dominant traits in human being, and these are some recessive traits in human. Let's see what is monohybrid crosses. Mendel selected a single trait and he crossed. So that cross is called as monohybrid cross. So what is this in the sense? He took the yellow seed and he crossed with the green seed. So here he has taken the pure yellow in the sense the Alleles are same alleles. So homologous. Here it is heterozygous. Here it is homozygous. Here it is here is also homozygous, but this is recessive and this is dominant. This one is dominant and this one is recessive. Okay, dominant is yellow and recessive is which express the green color. The gamete formation, this possess this gametes possess capital Y, this gametes possess small y. While crossing these two, we get a hybrid in F1 generation. We get a hybrid. What is that hybrid? In the sense it is yellow color in phenotype, but in genotype, if you see, it possesses one capital Y and one small y. That is heterozygous. Okay, heterozygous, and they are yellow in color. So all the hybrids after crossing this yellow and green is yellow only. So he didn't understand. So after he crossed these two, that is F1, okay, with the capital Y and small y. After that, he found it out. What is the result in the sense? One type of gametes possess capital Y and other type of possess small y. So when crossing takes place between these two in the sense, what happens? There are 3 yellow and 1 green. How come? The phenotype, one is pure, that is one is homozygous, and the other two are heterozygous, and one is homozygous recessive. So he got 3 yellow and 1 green, whereas here all yellow, whereas in the F2 gen he got only 3 yellow and 1 green. Okay, so what is phenotype and what is genotype in the sense? Phenotype is external traits are called as phenotype, and internally the gene representation, the gene present and what gene is present inside, that is inside the chromosome or inside the cell, that is called as genotype. So the organism underlying genetic makeup, which alleles it is called, its genotype. So what type of alleles present in the particular gene or particular chromosome, that is called as genotype. Okay, the observable traits expressed by an organism are referred to its phenotype. Okay, now we can see dominant traits. What do you mean by dominant trait in the sense? Dominant traits are those that are expressed in hybridization. There is a two different type of or two different genotype plants are crossed in the sense it gives rise a hybrid. Okay, the process is called as hybridization. So the trait which is expressed during the hybridization, that is called as dominant trait. That is, we crossed yellow and green. What we got in the hybrid in the sense, we got yellow. So this character or this trait is dominant. That is called as dominant trait. And recessive trait, what is recessive trait in the sense? It become latent or disappear in the offspring of hybridization. That is, green color is disappeared completely. We got all the offspring are yellow. So the green disappeared there. So this is called as recessive trait, but they reappear in the progeny of the hybrid offspring. And then in the next gen, the green appeared, isn't it? Green color appeared. So that is recessive traits. So recessive traits become latent or disappear in the offspring of hybridization, but it reappear in the progeny of the hybrid offspring. Next, what we are going to see is codominance. So what is codominance? The variation on incomplete dominance is codominance, in which both the alleles for the same characteristics are simultaneously expressed in the heterozygote. Whatever may be the allele, they both express simultaneously. So that is called as codominance. And multiple alleles. what are called as multiple allele in the sense one dominant and one recessive. One dominant and one recessive could exist for a given gene. Okay. Mental implied there is only two alleles, but multiple allele in the sense individuals like human and all other diploid organisms can only have two alleles for a given gene, but multiple alleles may exist at a population level such that many combination of two alleles are observed. One of the example is the blood group of human being. You can see here, if this comes, that is capital A, alleles come in the sense, this blood group is a A blood group. If it is A and B comes in the sense, it is AB blood group. And if it is one single A and this allele is recessive also, it expresses as A only. And if it is B and A, then it is AB. If it is B and B, then it is B. If it is one B and other is recessive also, we get B. And then only one A in the sense, and the next is recessive also, the blood group is A. And only one B and the other is recessive also, the blood group is B. If it is both recessive, then it become O. Let us see the multiple alleles in detail. Most of the genes exist in two alternative forms having different phenotype effects called alleles or allelomorphs, which occupy the same locus in the homologous chromosome. So where the allele present in the sense, they occupy the same locus in the homologous chromosome, the alleles. Okay. Usually a gene is represented by a most common allele, which is called as wild type, and an alternate allele, that is the mutant. Okay. However, there are several genes that exist in more than two alternative forms. Several genes that exist more than two alternative forms are called multiple alleles. If it is usually it is only two, but there are several genes that exist more than two alternative forms of multiple, so that are called as multiple alleles. Okay, which control alternative forms of the same character. They occupy the same locus in the pair of homologous chromosomes. And these multiple alleles controlling a particular trait may be present in a population of individuals, but only two alleles are present in a diploid individual. Example, we have seen ABO blood group system in man, as well as self-sterility in Nicotiana tobacco. Nicotiana means it is tobacco. Now let us see the human sex-linked disorder. What are the disorders are sex-linked? Sex-linked in the sense, it is the sex chromosome linked. Okay. So what is that in the sense? In Morgan laboratory provided the fundamentals of understanding the X-linked recessive disorder in humans, which included, what are those in the sense? Red-green color blindness, type A and B hemophilia, muscular dystrophy. The dominant lethal inheritance pattern is one in which an allele is lethal both in the homozygote as well as in the heterozygote. Okay, whether it is heterozygote or homozygote, the lethal gene, the dominant lethal inheritance pattern is one which is an allele is lethal in both. Okay, both homozygote as well as in heterozygote. And dominant lethal alleles are very rare because the allele only lasts one generation and it is not transmitted in the next generation. So an example, if you see the humans, that is a disease called Huntington disease, in which nervous system gradually wastes away. The onset of this disease may not occur until age 40. Make a note of this point. It may not occur until 40, until the age 40, at which point the affected person may have already passed the allele to 50% of their offspring. Okay, these allele were passed 50% to their offspring. So that is the thing in this disease, that is Huntington disease. You can see here, this is the neuron. At the center you can find there is a yellow color which has a nucleus, which has a nuclear inclusion. It is the characteristics of Huntington disease. This orange color area, what you are seeing in the center of the neuron, orange color area at the center of the neuron, what you are seeing here occurs when an abnormal dominant allele for the Huntington gene is present. Okay. And another one more we can see. The Y chromosome consists a small region of similarity in the X chromosome. So that what you are seeing here is the Drosophila. This is Drosophila, and it possesses the white eye as well as the red eye. How this is in the sense it is sex-linked or X-linked. So what is called as X-linked in the sense? You can see this chromosome, that is the Y chromosome. Y chromosome contains a small region of similarity to the X chromosome, so that during meiosis they pair. Okay, this X chromosome and Y chromosome has a small region just like that of the X chromosome. That's why they pair during the meiosis. Okay, the Y chromosome is much shorter and contains many fewer genes. When a gene is present on the X chromosome, it is said to be X-linked. Okay, the disease causing or any other gene which is present in the X chromosome in the sense, and this is passing from one generation to another generation in the sense, then those are called as X-linked. Okay, see one example we can see here. It is the eye color in Drosophila. I told you there are two eyes, one is white in color and another one is red color. Okay, so two different eye color we can find it in the Drosophila. So this is X-linked trait. Okay, it is just like human. The Drosophila males have XY chromosome and females are XX. Okay, so implies the wild type, that is the red color X capital W. This chromosome is responsible for, okay, this gene is responsible for red color eye, that is this one, and X small w, X capital W is responsible for red color and small w is responsible for white color. Okay, white eye. This is responsible for white eye. The females can be, okay, females can be X capital W, X capital W, and X capital W, X small w, and X small w, small w. The female can be in these three genotype. Okay, these three genotype are female, whereas in male there is only what we call two types of, there are three types of genotype in female, whereas in male there is only two types, that is X capital W and Y, and one more, or it is X small w Y. Okay, it may be either in this form or in this form. So only one type the male is, whereas in female there are two types. Okay, either this one or this one. So another one important thing is hemizygous. So these males are said to be hemizygous because they have only one allele for any X-linked characteristics. Okay, so they have only one. Here you can see there are two X-linked. So 2X. But here there is only 1X. Okay, so this is called as hemizygous because they have only one allele for X-linked characteristics, whereas in female there are two X-linked characteristics. Okay, two X-linked chromosomes are there in female, whereas in male there is only one. Okay, so this hemizygosity, which makes the description of dominance and recessiveness irrelevant for XY males. And what you are seeing here is a crossing over. The chromosome is crossing. So this bit come along with this one. Okay. And this bit is coming to this side. Okay. See, this is red color. And here the detach takes place here. So this part comes to this chromosome. So you can see clearly here, the small c comes here and the capital C goes there. This part is exchanged during crossing over. This is happens in the meiosis, though, so by the variation in the animals. Okay. So another one important thing is malaria is a parasitic disease that is transmitted to the human by the infected female Anopheles gambiae. Okay, this mosquito is transferring this malarial parasite. What is that parasite in the sense? It is Plasmodium falciparum. Very important concept we are going to see is epistasis. What is epistasis in the sense? It is the interaction between the genes is antagonistic, that is one gene masks or interferes with the expression of the another. That is what called epistasis. What we are seeing here is there are three traits. One is agouti, one is black, and another one is albino. So they possess two pair of alleles are here. So if it is come in the sense, it is agouti, which is dominant over the solid color fur. Okay, so if the C comes in the sense that gives a solid color, it is black. So the C is responsible for the formation of the pigment. So you can see here, one C alone is enough to bring the solid color. Okay, which is black. How many are in the sense? 1, 2, 3 are there. And if the C comes in recessive, then it becomes albino. Okay, it becomes albino. And this chromosome, this with the A with CC, capital A with CC, that form the agouti. Okay, one capital A and one capital C alone is enough to form the agouti. You can find here. So one capital A and one C alone, they form the agouti. So for the formation of the agouti, it needs the C and A dominant gene, and the other, which one? The C is responsible for the solid color, that is black. Okay, so here what happened here, what you are seeing here, one gene is masking, or one gene is interfering the expression of the other gene. Okay, that is what this experiment is explaining. And this agouti, how many, see, if we are crossing two agouti in the sense, these are the result we are getting. So what is the gene which is in that, in the sense, capital A, small a, capital C, and these two are hybrid of F1 generation, then it is crossed, in the sense we are getting these many. So in that 9 are agouti, 3 are black, and 4 are albino. That is 9:3:1 ratio. The F2 is 3:4 ratio. The F2 is okay. So this is called epistasis. So interaction between the genes occur here. Okay, in antagonistically, that is one gene masks and or interferes with the expression of the another one. Thank you. Thank you so much. Share this video to your friends, those who are preparing for CSIR NET. Thank you. Thank you so much. Share and subscribe this video. Thank you.